Good morning, students. This is Manoj Kumar Nalla. Today's class is mutations. What is the mutations? Sudden irritable change in genetic material or character of an organism known as mutation. Individuals showing these changes are known as mutants. An individual showing an altered phenotype due to mutation are known as variant. Factors or agent causing mutation are known as mutagens. So, sudden changes in heredity character, sudden changes in genetic material. Due to these change in genetic material that shows a different in phenotype character, that is called mutation. So, the individual shows mutation are known as mutant. So an individual showing an altered phenotype due to the mutation are known as variant. So causing agents, mutation causing agents are known as mutagens. Mutation which causes changes in base sequence of a gene or an gene mutation or find mutation. History. English farmer Stith-Wright recorded cause of mutation first time in 1791, male lamb with unusual short legs. The term mutation is coined by Hugo de Vries in 1900 by his observation in Enothera lamarckiana. Systematic study of mutation was started in 1910 when Morgan genetically analyzed white hive mutant of Drosophila. H.J. Muller induced mutation on Drosophila by using X-rays in 1927. He was awarded with the Nobel Prize in 1948 for his work on mutation in Drosophila. Characteristics of mutations. Generally, mutant alleles are recessive to their wild or normal alleles. Mutant alleles are recessive to their wild type or normal alleles. Most mutations have harmful effect, but some mutations are beneficial. In nature, most of the mutations have harmful effect, but some mutations give some beneficial to organisms. Spontaneous mutations occurs at very low rate. So the mutations in spontaneous occur is very low rate. Some genes shows high rate of mutation. Such genes are called as mutable gene. Highly mutable sites within a gene are known as hotspots. Mutation can occur in any tissue or cell of an organism. Classification of mutation. Based on the survival of an individual, mutations are the following classic type. One is the lethal mutation, sublethal mutation, subvital mutation, vital mutation, supervital mutation. So based on the survival of an individual. Lethal mutation. When mutation causes death of all individuals undergoing mutation are known as lethal. Sublethal mutation causes death of 90% individual. Subvital mutation. Such mutation kills less than 90% individual. Vital mutation. When mutation don't affect the survival of individual are known as vital mutation. Supervital mutation, the kind of mutation enhance the survival of individual, that is called supervital mutation. So based on the survival, lethal, all individuals are causes death. Sublethal, death 90% individual. Subvital, less than 90% individual in death rate. Vital, don't affect on survival. Supervital, enhance in survival rate, that is the supervital mutation. Based on causes of mutation, the following types of mutations are there. 1. Spontaneous mutation. Spontaneous mutation occurs naturally without any cause. The rate of spontaneous mutation is very low. Example, methylation followed by deamination of cytosine. Rate of spontaneous mutation is higher in eukaryotes than prokaryotes. UV light of sunlight causing mutation in bacteria. Induced mutation. Mutation produced due to treatment with either a chemical or physical agent are called induced mutation. The agent capable of inducing such mutation are known as mutagen. Use of induced mutation for crop improvement program is known as mutation breeding. Based on tissue of origin, mutations are two types. One is the somatic mutation, second one the germinal mutation. A mutation occurring in somatic cell is called somatic mutation. In sexually reproducing special somatic mutations, one transmit progeny to the next progeny. Germinal mutation. When the mutation in gametic cells or reproductive cells are known in germinal mutation. In sexually reproductive species, only germinal mutations are transmitted to the next generation based on the direction of mutation. Based on the direction mutation. Forward mutation, reverse mutation. When mutation occurs from the normal or wild type allele to mutant allele are known as forward mutation. Reverse mutation. When mutation occurs in reverse direction, then is from mutant allele to the normal or wild type allele are known as reverse mutation. Type of trait affected, visible mutation. Those which affects phenotypic mutation and detected observation are known as visible mutation. Biochemical mutation. Mutation which affect the production of biochemicals and which does not show any phenotypic character are known as the biochemical mutation. Chromosome mutation may involve change in the structure, loss or gain. Chromosome mutation. 5 types of exist. Deletion, inversion, translocation, non-disjunction, duplication. Deletion. Due to breakage. Deletion causes due to a piece of chromosome is lost called deletion. So, in that piece, a loss of piece of containing some genes. Some genes loss of organism, it shows some different character. This is the deletion. Next one, inversion. Chromosome segment breaks off, segment flip around backwards, that is called inversion. To see this map, this is the part break of chromosome and inversion and attached attached reverse, that is called inversion. Duplication occurs when a gene sequence is repeated, that is the duplication. So the genes are repeated. The repeated genes causes some character, that is the duplication. Translocation. In translocation, 2 chromosomes are involved that are not homologous. Part of one chromosome is transferred to another chromosomes. So here see, before translocation, AB chromosome, between the AB chromosome, the part is transferred from to one chromosome to other chromosome. This is the translocation. Non-disjunction. Failure of chromosome to separate during meiosis, that is called non-disjunction, causes gamete to have too many or too few chromosomes. So, due to the disjunction, the organism, the progeny organism, loss of one chromosome or few chromosomes. So, disorders due to non-disjunction, Down syndrome, Turner syndrome, Klinefelter syndrome. Chromosome mutation. So, A, B, C, D, E, F, G, H, I. This is a sequence. In this sequence is difference is there, that is the chromosome mutation. So types of gene mutations. So based on the chromosome gene sequence altered, that is called gene mutation. Gene mutation types is the point mutation, substitutions, insertions, deletions, frame shift. Point mutation, change of a single nucleotide include the deletion or insertion or substitution of one nucleotide in a gene. The changing occur, the gene sequence is changed. That is called uh, point mutation. For example, sickle cell anemia. In the result of one nucleotide substitution occur in the hemoglobin gene, that is called point mutation. So, due to the change of the nucleotide, glutamic acid replaced by valine, uh, change the sequence of the nucleotide, the amino acid change in the hemoglobin, that is the glutamic acid replaced by valine, that causes sickle cell anemia in humans. Frame shift mutation, inserting or deleting one or more nucleotides, changing the read frame like changing of sentence, proteins built incorrectly. Gene mutation, CGAACDCCA, this is the sequence. In this sequence, changes occur, that is called gene mutation. This is the gene mutations. Thank you very much.